All right, 12:31. Let's start. You guys can see you guys can see the PowerPoint screen as well.
Yes, Professor.
All right, great. All right, so this will be our last module 4.2 part 1, and I will divide this. I have divided this into 3 parts, part 1, 2, and 3. And what I will do that I will actually upload the part 2 as well as part 3, which we are going to talk about in the next 2 classes by this weekend. So you will be able to see the part 2 and 3, and I will try my best to upload the videos as well, so that you just do have still 1 week in buffer to look at the material, and you will be in the comfort zone. And again, I have cut down a lot of material, and you will see that the material is not difficult. Make sure that you study, start studying early for the final, and again, you will have all the chances to improve your grade even better. And so far, what I have seen that there are 2 questions that were wrong. It was not confusing. Both of the questions are actually, when I read that, my key was wrong. When I just transferred them from a Word file to the to my question bank, these things happen all the time. So 2 questions are going to be thrown away, in the sense that, not thrown away, I will just add 5 points to the bonus column. So you will see that in the bonus column, it is now standing at 17.5, and you will see that from that, it will jump up to 22.5. So 5 points added to your existing scores at exam weight. So that's going to happen. Any questions regarding that, anyone? No? If you have any questions, let me know. I will be more than happy to. Let me open up the chat window as well, if you have any issues with that. Hold on. Why it is causing a problem. Okay, all right. So it's all good. So we're starting with the cancer cell biology. And the first thing that why we are talking about cancer at the end, that there are 2 things that you will see that many of the things that we have studied in this cell biology course, they will all come together, and there is a lot of synthesis of the material, which will explain that how we study the cancer cell biology. And along with that, it is really important that 1 in 5 individual of the prosperous countries worldwide will die of a cancer. And you know that everybody is touched by cancer in some way or the other. Whereas it is not that relevant, interestingly, in some of the developing, not some, many of the developing countries or the third world countries, because we are going to see that there is a statistics that usually malnutrition or infectious disease and other diseases are main cause of cancer. So far, if you just pull out from, if you just go to CDC and pull out the data, that the number of people each year die due to different diseases, still, as of now, the data of 2018 is available. Heart disease is the number 1 killer in the United States. Now, cancer is number 2, but it's very, very close, very, very close. And probably in the next few years, cancer will supersede the death by heart disease. And we will explain that why this is. So what is basically the cancer? So in young animals, when they are at a growing stage, you can see that the cell division, the amount of cell division way exceeds the cell death. And this happens very, very fast in a newborn to the fetal formation. And then gradually as we age, this cell division and cell death comes to an equilibrium at a point where the cell division is equal to cell death in an adult animals, which we call as a steady state growth. On the other hand, this whole process is controlled, we have seen beautifully, by the cell's exquisite control of regulating the cell multiplication. So the cell cycle basically drives this whole process, whereas the cancer is basically a breakdown of the cell's exquisite control over this cycle. And the cell grows and divides in an unregulated fashion, and that is basically the cancer. And we're going to use some of the terms. A tumor cell is also known as a transformed cell, and they are also called the neoplastic transformed cells. Plasticity, we have already talked about that. So that's basically what it is, that the normal cell, how it should differentiate from that norm, it is now gaining some plasticity to become a new type of cell, and this is a cancer cell. So we will use this term neoplastic. And the cancer cells shows something which is called a neoplastic phenotype. And again, if you just remember from your Bio 101, what is a genotype is something that is changed at the genetic level. That happens, and due to that, you have this neoplastic phenotype that you see in the cells and outcome of the tumor that you can see. So phenotypes that you can see visually, and we are going to dissect the genetic cause or the genotype, what caused this as well. So that's an overview of your cancer. So here we are looking at the tumor types, and there are two types of tumor. So the first type is a benign tumor. We all know this. And remember that even in the benign tumors, that is still uncontrolled cell growth. And these cells will be called neoplastic cells as well. But still, this is better in the sense that these tumors remain localized, but they only become dangerous if bulky or biologically active, and sometimes if they secrete hormones. But there are some exceptions to that, and we will come to that in a second. So these are, generally speaking, the benign tumors are not a huge problem. And you can see that the benign tumors, as simple as a skin tag, can be considered as a benign tumor. On the other hand, if we just talk about the malignant or the metastatic tumor, these cells, these are also neoplastic cells, similar to that there is some problem with the regulation of the cell cycle. So they will grow in a way, uncontrolled way, but they will not be confined to this fibrous connective tissue. They will be moving into different tissue types, which we call as a metastasis, and they grow far more rapidly than these benign cells. And this is the dangerous and the life-threatening cause of cancer. What is it's all about? So here, what you are seeing, that if you're just interested, that what is that? So here is adenoma. This is a very specific type. So this adenoma is usually the epithelial glandular structure. If they form a tumor, we call this adenoma. And adenocarcinoma is the cancerous form of an adenoma. And sometimes these are as simple as, we call this as a polyps or something like that. So just think about that sometimes when you just do your screening of your colon, usually which is recommended above the age of 50, sometimes you probably have seen the doctor say that there were some polyps that were removed. So sometimes we grow these polyps inside our intestine, and when we do the colonoscopy, these polyps are visible, and these are usually benign tumors. But the doctors actually do remove those, because not the reason that they will become dangerous or bulky, which very well they may be, but chances of these cells to become cancerous is much more compared to a normal cell. So just to make sure that these benign tumors do not convert in later into a cancerous tissue, we remove those. So this is just an overview of idea that what is a benign tumor and what is a malignant or the metastatic tumor. And we are only going to talk about this part mainly. But remember that both of them, that there is some problem with the cell cycle, and both of these phenotypes are the neoplastic cells. And just these are much more invasive and much more dangerous compared to these. So let's take a look, and this is a revisit from your first chapter or the first module. So cells, when we grow in this culture, we already know that we need this essential amino acid, at least 9 of them out of 20, vitamins, growth factors, which comes from the serum that is being supplied. And most of the cells require a solid surface, means they are anchorage dependent. So if you take a Petri plate where you are culturing your cells, or a tissue flask, any kind of vessels, the cells will be attached to the bottom. So this is what is called is anchorage dependence. And also they show another phenomena, which is called as a contact inhibition, means that if another cell touches to each other, that will stop the growth of these cells. So what happens that when we culture these cells in the media, in the laboratory, once they are confluent, that means they form a monolayer almost touching each other, it is time to passage those cells. That means that you divide those and transfer a small number of these cells to a new flask or a new Petri plate to allow them to grow, because once they touch each other, they will not grow and gradually they will start dying. So this is one of the things. But there are some exceptions. You remember that many of these cells, there could be two different types. One is a primary cell type, which does have a finite number of growth cycles. So they will divide 50 to 100 times and then they will die off. And there are lots of cells which are derived from the cancerous tissue, which are called the immortal cells. They will divide indefinitely and they may not have the contact dependence. They may not have the inhibition, contact mediated inhibition of growth. So they can grow in piles, even they can grow in suspension cultures. And we utilize these to produce many different products which are grown in animal cell culture, because you can grow a much larger number of cells if you are growing them in the non-suspended media, like in a fermenter, that kind of flask. So let's take a look at what happens to a cancer cell. So if you just look at these cells, these are the fibroblast and these are the scanning electron micrograph. And think about that. These are the normal cells. These are not neoplastic cells, and they are growing on the surface of the vessel. And in a cartoon, they will look like this. So they are contact inhibited monolayer of normal cells. But if you just look at these neoplastic cells, which are transformed, now you can see that this cell is on top of this cell is on top of this. So they are growing on top of each other, which is like this. So they are not contact inhibited and they will pile up against each other. So why this is so? And we are going to talk about that in a moment. So let's take a look at that some changes that are commonly observed in the transformed cells. So the first is the adherence abnormalities that we just talked about. So the first thing that we talked about is the diminished adhesion to the surface, thereby able to maintain this rounded morphology. These are colored scanning electron micrographs. So scanning electron micrograph, but they are pseudo-colored, like artificially colored. But you can see that they're growing on top of each other. And here is an important thing just to understand, that failure of the actin filaments to organize into stress fibers. What happens that if you just go back to this particular cell right here, the fibroblast, near the substratum, which is basically the bottom portion of the flask or the tissue culture vessel, you have these stress fibers. These stress fibers are made up of actin, and that basically makes the cell keep the cell into an integrated format, because what is happening when the cells are stretching to make sure that the cells are not tearing apart. So these stress fibers are not organized. So the actin filaments in the cancer cells are very disorganized, and that's the reason they've become rounded up. Another thing is that outside these cells, there's also a coat of fibronectin. So this fibronectin is an ECM, or extracellular matrix protein. And when you have this fibronectin, this is an attachment protein for these cells into the extracellular matrix. And they naturally will have that. So now if we just go back to the benign tumors picture right here, you have the benign tumor picture and you have this fibrous connective tissue. So the benign tumors still have this layer of fibronectin surrounding them to which they are attaching right here. But in the neoplastic cells, that what we are looking at, this fibronectin layer or the fibrous connective tissue is even gone. So that's the reason that they can migrate out of their space to other places. That's been shown here, fibronectin. Then there is another interesting thing, that these cells produce something which is called a plasminogen activator. So again, we will not go into the detail of the whole procedure, that what it does, but this plasminogen activator will activate a particular protein in the extracellular matrix, which is called plasmin. And this plasmin cause increased extracellular proteolysis. So this is not a normal condition. And this basically what happens, that this is related to blood clotting and other things, but this is a particular phenomena where these cells will produce these plasminogen activators. So outside these cells, there will be a large amount of protease or proteolysis activity. And what it will do, that it will digest the protein fibers in the extracellular matrix, which are responsible for the attachment of these cells. This is a very important concept. Again, repeating, this plasminogen activator will activate plasmin, which will increase the proteolysis or proteolytic digestion of the extracellular matrix protein, which will inhibit the attachment. So these extracellular matrix proteins are responsible for the attachment of the cancer cells, but if they're degraded due to this plasminogen activating plasmin, then that attachment will not happen. So this is a particular way that how this protease activity help them not to attach onto the substrate or the extracellular matrix. So that's your adherence abnormalities. The next one is, of course, that we know that the growth and division abnormality. And here, the growth to an unusually high cell density, that there is no contact inhibition, lowered requirement for growth factors. So we have already seen that the normal cell, you need the serum, which is full of these growth factors. Those are even not needed for the cancer cells. And loss of anchorage dependence can grow even without the attachment to solid surface that we have just seen, and loss of contact mediated inhibition. So what it does, that due to that, they will pile up on top of each other. And here you can see that right here, these are the cells still are in a monolayer. So they are still single towards the periphery. But here you can see that they piled up, and it almost looks like a three-dimensional image in this phase contrast microscopy. So you can easily see that this is a little bit elevated because they are piled up against each other. And this is called a focus or plural foci. And of course, the cells are immortal, can continue proliferating indefinitely. So we just talked about at the very first chapter about a book, which is about the Henrietta Locklear in 1950s, late 1950s, a cell from her cervical tumor called HeLa cells, which is after her name, was isolated. And that is still used in the number of different laboratories. And that is the cell that is used by the scientist, probably the most used cell in biomedical science, cell biology, or any kind of thing. So still it is growing in thousands of different laboratories across the world. And the cell was originally isolated from a tumor in 1956. So think about that. So they are immortal, continue to proliferate indefinitely. And if you take these cells, which are the neoplastic cancerous cells, and if you inject these cells to susceptible animals, they will form cancer. What we mean by the susceptible animals, it will not cause cancer to everyone. But because what happens when you take these cells and put into another animal, if the animal does have a robust immune system, it's immediately going to attack these foreign cells and kill them. So if the animal is immunocompromised, that is what we mean by susceptible animals, and that will cause this, this particular tumor formation in that. We're going to talk about that. All right, so today we have just only one clicker question because it is a little bit food for thought, and we will have more questions in the next two chapters, which will cover some of those. So we'll have 3 each in next 2 sessions, but today we have just only one question. So right here, to remove the cells from a tissue culture flask, this is from your first chapter, EDTA and trypsins are used. Which of the following neoplastic phenotype of a cancer cell you may consider as analogous to trypsin? And again, it is probably a question which you need to remember your first chapter, And this type of question will not appear just by themselves in your exam. I'm just giving you this example so we discuss, and that's why this may appear as such. So absence of actin stress fibers, reduce cell coat by fibronectin, high production of plasminogen activator, release of excess calcium in the extracellular matrix. Good, everyone? I'm stopping it now. Make sure you press your button. Anybody good? Everybody good? Okay, I'm stopping it now. All right, so 55% right here, the high production of plasminogen activator. So let me explain that why this is so. So if you remember that, think about that what trypsin does and what EDTA does. And again, this is the first chapter. If you do not remember, I completely understand. What EDTA does, that this is a metal ion chelating molecule. So if you can correlate that calcium is needed for the binding, and now you know why from your previous chapter, because calcium is needed for the function of the cadherins. So if you take out the cadherins from the CAMs or the cell adhesion molecules, EDTA is responsible for that. So EDTA will remove the calciums. That's its function that you don't find here. But we are asking about the trypsin. Think about what trypsin does. Trypsin is a proteolytic enzyme which will digest the extracellular ECM proteins or the extracellular matrix protein by protease action or proteolysis. So trypsin is a protease. Now, if we just go back to our question right here, high production plasminogen activator causing increased extracellular proteolysis. So the exact function, what trypsin does, a cancer cell, just by producing plasminogen, will make this analogous to trypsin. And again, this I will not ask without you being told this, because otherwise this is an information from your first chapter, and I do not expect that you will remember that to answer this question directly from that. So everybody good? Any questions on this one? If you need any explanation, I can explain that. So this will, I'm just telling you, this will be exactly this way in your exam. So you already know one question. I will put that one in because you guys are coming to the class, everyone. So you will just get the benefit of it. So good? Okay. All right. So let's go to the next one. So right here now, let's go back to the previous idea that the scientist, and this is like a long back. So basically the scientists are now thinking that long back, that what caused this cancer. So this is basically the discovery of the cancer, what causes it. So what the scientists did, that they thought that was a hypothesis at that time, that there must be something wrong in the DNA that probably caused the cancer. And they did some experiment, which we call as a transfection assay. You isolate a tumor. From there, you isolate the DNA from those tumor cells, the genomic DNA from those. Then you take some of the mouse fibroblast. These are the primary cultured cells from the mouse fibroblast, and you transfect these cells with this DNA. And you can transfect by many different means. You can use a liposome-mediated transfection or electroporation. There is lots of ways to put these DNA inside the cell. You can easily do that. That's not a problem at all. Lots of different means. So what will happen, that these cells will grow in piles, some of these, not all, where you successfully transfect this DNA. And these are the transformed cells. These will form these foci, which are the neoplastic phenotype. Now, if you take these pile of cells from these foci, the transformed cells, and inject to a mice, and this is a special type of mice, you will see that there is a formation of this tumor. So why this is so, and what is happening here? So the important thing is that means that the scientists understood that there must be something in the DNA of these tumor cells that is causing that cancer. which is from the tumor. Now, if you remember that we used a word, and I will just explain that here, that right here, can cause tumors when injected to susceptible animals. And here, what you are seeing, that this is a mouse which is susceptible. And there are many varieties of that. Some of them are called a nude mouse. This is a skid mouse on the other hand. So there are differences. The nude mouse doesn't have any T lymphocyte, which have a there is no hair on the skin as well. The skid mice looks the same, but it is a little bit different than the nude mice. The skid mice is called the severe combined immunodeficiency syndrome. They don't have T cells as well as B cells. So they do not have any lymphocyte. So there will be no immune cells of the T and the B cells, no cytotoxic T cells, and that will not be able to kill these foreign tumors. That's why it will grow. If you inject these transformed cells into a normal mouse, which is not immunocompromised, the immune system of the mouse will immediately attack these transformed cells as a foreign origin, and they will kill them. And still, chances of tumors are possibility, but it will be much less. So that's basically what the experimental protocol is. So take-home message from there. Now we know that there is something in the DNA and what it is. So now we are going into something which we called as that what is the basis of cancer. And the first basis of cancer was understood not long time ago. This is 1989. Michael Bishop and Harold Varmus, they actually isolated and described the process of cancer. Now, remember that this was not the first oncogene. We will come to these definitions, not the first oncogene that they discovered, but they discovered the mechanism by which an oncogene can cause a cancer. And they discovered this. I talk about this a lot in my virology class. So the ones who are from my virology class, we talked about a lot. So they basically understood that there is a gene called cellular SERC gene. Not going into the detail, SRC or SERC. So this particular gene is picked up by a retrovirus. And in the retrovirus, it converts it to a mutated form, which we call as a viral SERC or VSERC. Now, when that particular retrovirus infects another mammal, it injects or basically input that VSERC into the genome. Because remember that a retrovirus will integrate its genome just like HIV into the genome. So that VSERC is an oncogene, will cause a cancer. So that they discovered in a Rous sarcoma virus, which is a cancer-causing virus. So that was the Nobel Prize. And I just described that why this is a monumental discovery, because they received the Nobel Prize in 1989 for a work in 1977. And I just discovered that the time when the scientists discovered something monumental and they were awarded a Nobel Prize, the time span in between them tells that how important is the discovery. So anyway, so after this discovery, there are some of the specific thing, and there were many, many genes after that were discovered in that process. And we're going to define some of these which cause the cancer. So we have some genes in our body, which again, before going to the definition, we do have a number of genes which play a role in normal cell growth and proliferation. And these are called a positive regulator of cell cycle. I'm just giving you, before the name, just giving you an example. Suppose you have your epidermal cells, epithelial cells, skin cells. If the skin cells get a stimulus from outside, there are some specific receptors are there on these cells which will tell the cell to divide. This is constantly happening to your intestinal cells, which is constantly dividing. So these are some of the positive regulators which control the cell cycle or make the cell cycle go on, provided that everything is good. So these are called the proto-oncogenes. This has nothing to do with cancer. These are just the normal cell growth and proliferation. So the name is a kind of a misnomer, and I'm going to just explain that why it is so. So now what happens that if this normal cell growth proliferation control, which is working every day in our cell, have a mutation, which means these proto-oncogenes are mutated, they become oncogenes. So they gain a function, and this gain of function mutation contribute to transformation of cell. So that's basically what it is. And these are dominant oncogenes. So your proto-oncogenes, when they form oncogenes, they become the dominant. So what happens that if you have two alleles in your chromosome. So if you have just only one of them is an oncogene, you will have that as a dominant. So that means that if you have the mutation in both alleles, of course, that is homozygous and it is dominant, it will cause cancer. If you just have one allele, like with this mutated oncogene, the other allele is still okay, which is a proto-oncogene, the heterozygous condition, still you will have the cancer-causing gene activated in you. It is only the possibility that you will not have when both of them are in the recessive format, means that none of them has mutated into an oncogene. So I'm just giving you a very crude example. So think about this, that how this basically works. So think about that you just do have a light bulb outside your house and you have a corresponding switch to that. Now that light bulb is present there all the time. So these proto-oncogenes are present there all the time. Now in the night when it's dark, you turn on a switch and that light bulb comes on, gives you light. So the same way that a proto-oncogene, if it receives a signal that the cell needs to divide, it will turn on the switch, which is the proto-oncogene, which is a positive regulator, and the light bulb will be on and the cell cycle will go on. Other time it is off. When it is converted to oncogene, means you have a short into your switch. The light bulb is on all the time. It is not under the control of that switch that you can turn it off. There is some short happening inside your conduit or whatever it is, that it is now constantly on. So that's basically the concept of your oncogene, that once you have this mutation, it's constantly on and there is uncontrolled cell division. That's what it is. On the other hand, the tumor suppressor gene is another set of genes, and the function of these is the normal function in the inhibition of cell growth and proliferation. These are the negative regulators of the cell cycle. So, and again, we will talk about that. You probably are thinking about the retinoblastoma and the other thing, and we're going to talk about that in the subsequent sections. But think about that, just giving an overview of idea of a tumor suppressor gene overview. There are some of these histone acetylases, some methylases. So the histones, when they are packing the DNA in a very tight form, they will not be able to replicate. And that will not lead to a synthesis phase, that will not lead to a cell cycle. So what happens that tumor suppressor genes will keep these nucleosomes in that tight format. But if they lose that function, the genes which are responsible for the cell cycle will now become activated, because the nucleosomes are not keeping the DNA in that tight form. They are now in the loose form. They are making expression of their DNA. They are replicating their DNA, and they are making the proteins responsible for the cell division when it is not needed. So what happens, that tumor suppressor gene is a loss of function mutation. That means the tumor suppressor gene basically keeps the cell cycle in check. It doesn't allow the cell cycle to go on. If there is a loss of function, then the cell cycle will go on into an uncontrolled growth. And this is what is a recessive mutation. So if you have two alleles, you need to have mutations in both of them in a homozygous form so that the tumor suppressor gene will be non-functional. If both of them are completely functional and normal, of course, you have the tumor suppressing activity. If they are, one of the allele is normal, another is mutated, which is the heterozygous condition, still that is a functional gene. On the other hand, if both of them are mutated to a loss of function, then it will cause or become a candidate for cancer-causing genes. Now, just remember that the oncogenes, tumor suppressor genes, their function is not to suppress tumor. Oncogene function is not to cause cancer. These are genes which are responsible for regulation of cell cycle every day or cell growth. When they gain the function, they become oncogenes to cause a potential tumor. When they lost the function, they become a potential cancer-causing gene. So they're a very important concept. So just make sure that you have a clear idea about this one. Any questions so far? For anyone? Good?
Don't think so.
Good? Okay. All right. Yeah, I mean, like, this is a very important concept, and that's why I just spent a good amount of time. All right, here is a very interesting idea. And again, we are not going to ask you any of these numbers in your exam. This is just an overview of that. So if you just think about that, how many number of cell division happens in the human lifetime, and of course, this is age dependent, but that does not mean that number will change that much. We will have about 10 to the power 16 cell division in human lifetime. I mean, like, it's a mind-boggling number. So at that time, the scientist also clearly can understand that how much DNA, once replicated by the DNA polymerases and the additional enzymes, how much mutations is the limit for our DNA polymerase. So they found out, and this can be really done experimentally, that about 10 to the power negative 6 mutations per gene. Remember that, not per base, not per ATCG, a whole gene, which could be several hundreds to several thousand base pairs, 10 to the power negative 6 mutation per gene per cell division can happen. But still, that number is pretty, it is a little bit scary. So what can happen in a human lifetime if you just do this, 10 to the power 10 mutations per gene in human lifetime. So that's a huge number, and we should not expect that, because we usually see that probably in prosperous countries, not even in the developing countries, we usually have about 20% people have cancers at certain stage of their life. So 1 in 5. So, but this number is really something very, very high, and you cannot correlate with the number of incidences. Why it is so? Because think about this, that for cancer to happen, you have to have in a same cell, think about that, not in different cells, in a same cell, at least 3 mutations, and as many as at least 7 mutations need to happen. And these are all independent genetic accidents. And again, this accident means it's a mutation. And if that happens, at least 3 to at least 7, then it can cause cancers. And that's actually a very good news in that sense. Otherwise, everybody will have cancer after a certain age. Another interesting thing, that why this is so and what is the validation of this data. So if you look at this particular data, epidemiological data, so you can see that this is the chances and the occurrence per 100,000 per people in, say, United States. So you can see that about 35, around 40, this curve goes up and almost exponentially as we age. So what is the correlation and how we come to this? Remember that, why this is so, because think about that, as we age, our cells are dividing more than we are. So as we are aging, our cells are dividing more and more mutations, independent mutations or the genetic accidents are accumulating in the cells. And when 3 or 7 in between them in a single cell happens, that will lead to the cancer. But again, this is a very interesting idea, that this is a concern for all the prosperous countries where the life expectancy is very, very high, very unfortunately. But that's where we see a lot of cancers. And that's one of the reasons that all the developed nations are working towards the cancer drugs so much. We are going to talk about that in the last portion. So that's the concern. But we will just show you, this is not in your PowerPoint. I just added this, and I will just show you something which is very sad, actually. So let's take a look at this. This is a, so I'm just going to show you here. Can you see that chart, everybody?
Yeah.
Okay.
Yes.
So yeah, so look at that right here. So if I just click on the USA, population 3.29 million, 329 million. And if you can see that life expectancy is about 80.1 years, female 82.3, male 77.8. And this chart is from the US. On the other hand, I'm just looking at, just look at this right here. This is some of the different nation. And 53 years is the life expectancy. Mozambique, right here, 54. Zambia, 53. Somalia, 53.2. Afghanistan, 52.1. And you can just imagine that there are lots of nations where you have the life expectancy in their 50s. So if you just look at that, see right here, this is the life expectancy. So that's the reason that, as I said, that really these countries do not care of the cancer that much, because most of the people die here due to infectious disease and malnutrition. The developed nations right here, where you have the life expectancy, we see way more cancer compared to the context of the other parts of the world. So that's something that you can think about. All right, so that's basically an overview of that. Again, not a specific information that questions will come from. So here is an experiment, which is very important, that the tumor appearance in mice overexpressing 2 oncogenes. So what it tells us that you need more than one, which we talked about right here, 3 to 7 genetic accidents required. And here we are supporting that hypothesis. So here what we have, that this is a MYC, this is a proto-oncogene, mutated form is the oncogene, and this is a transcription factor. So if you have this transgenic mice, and how this experiment was done, I just described it a little bit. So you take some mouse and then you introduce these transgenic mutations to that. And MYC, there could be many. It is not described in your book that what it is. RAS is a V12 mutation specifically. So what do we mean by that? So at the 12th position of the RAS, or the 12th amino acid, it's a glycine is a normal amino acid at the 12th position. So it is replaced by a V-valine. So glycine to valine, one amino acid converts the RAS, which is the guanine nucleotide exchange factor, into an oncogene, and oncoprotein will result from that. So what is happening here? So a mouse, you take a transgenic mouse and you just put a promoter in there, and you can turn on that promoter by certain means, that you just use a viral promoter which will respond to certain kind of hormone, and you introduce these transgenic these gene mutations into a particular tissue. It's a mouse mammary tissue, not all the cells. So now what happens that these promoters were the viral promoters, and they are hormone induced. So a very common hormone is your prolactin, which helps in the developmental of the breast tissues in the lactating animals. So you just add prolactin into the cells, or just add, exogenously feed the mice with prolactin, and that will cause these genes to express these oncoproteins. That was the experiment. But again, the experimental detail is not needed. The idea is that once this MYC is just being produced just by itself in the oncogenic form, you can see that the tumor is expressed about 125 days, tumor-free up to 125 days, then tumor happens. So you can easily see that the MYC is a kind of a weak oncogene. So the mice were cancer-free for quite a bit of time. On the other hand, if you see the RAS right here, this V12 mutations is a way more aggressive mutation in terms of cancer causing. You can see that right here, probably 40 days that mice were cancer-free or tumor-free, and then you can see that the development of cancer happens rapidly after that. But see that what happens that if you have a mice where you have the MYC as well as RAS both of them are mutated into the oncogenic form, you can see that how rapidly it causes the development of the tumor. So the experiment shows that multiple mutations do have a synergistic effect. So that's the word, does have a synergistic effect or additive effect, so that they can, both of these can increase the amount of tumor formation. So that's basically the overview of the synergistic effect of cancer causing condition. And as I said, that even this is an experimental technique, but in reality, at least 3 to 7 independent genetic accidents are required so that cancer appears. So this is in the support of that hypothesis. So lastly, what we're going to talk about, that proto-oncogenes and the tumor suppressor genes, and we are just describing the class. And in the part 2, we are going to talk about these proto-oncogenes. And in part 3, we will individually talk about the tumor suppressor genes. So what are these? So the proto-oncogenes are called class 1, 2, 3, and 4. And the tumor suppressor gene, there are 3 classes, 5 through 7. So what are these? So the first one is a signaling molecule. I will just give you a just brief example of here. You do not need to remember the examples because we are going to talk about the examples in much detail in the next portion. So signaling molecule, and if you just, we are not going to consider this though, but if there is a signaling molecule which binds to the receptor and then it activates the receptor more than it should, or keep bound to the receptor more than it should, it could be a problem. Think about that. You have an epidermal growth factor. That's a signaling molecule, EGF growth factor, that binds to an epidermal growth factor. And somehow what it does, that it just it is bound in there. It doesn't allow the receptor to get degraded, and it is just constantly signaling. So the cells are proliferating in the epidermis constantly, cells are dividing. So that can cause neoplastic phenotype. On the other hand, the second type is the signal receptor. If there is some mutations in there, and we talked about this, think about that, your receptor tyrosine kinase is a classic proto-oncogene. If somehow there is a mutation in the RTK, or the receptor tyrosine kinase, where it's constantly signaling, and that can cause the cancer as well. So often a protein tyrosine kinase. So you can remember the structure. The third type are called the intracellular transducers. So think about that. If we are talking about the tyrosine kinase, your intracellular transducers are your RAS, then your RAF, then you have your MEK, then you have your MAP kinase. So all these proteins which made these cascades, and all these proteins, if they are mutated in such a way that they will keep signaling the downstream pathway constantly, and if that downstream signaling is for cell proliferation, then that will cause cancer. So these are called the intracellular transducers, which are the signaling pathway proteins downstream of the receptor. The second type are also intracellular receptor. But remember that if you have a receptor on the plasma membrane right here, they are usually hydrophilic molecule, which are water soluble, because remember that extracellular space, this is all watery environment. So these are water soluble molecule, hydrophilic molecule. On the other hand, if it's a hydrophilic molecule, they can easily pass through the plasma membrane, and they will be bound to the intracellular receptor. And these intracellular receptor mutations can also cause cancer. This is another type, which is also type 2, same way. Both are receptors, remember that. Just the 2 different types of receptor, intracellular versus extracellular or plasma membrane receptor. So the next type, which is the third, we already talked about that. And then 4 is the transcription factor. So we talked about these transducers like your MAP kinase. Think about that, that you have many of these substrate of these MAP kinases, TCF, it phosphorylates the TCF and TCF transcription factors. There are serum response elements. All these are the transcription factors which drive expression of genes. And if these transcription factors do have a mutation and they drive expression of genes which are responsible for cell growth or cell division, that can cause cancer. So again, remember that all these 1 through 4, if you have a gain of function, that means that when it should not be turned on, they're on at that time wrongfully, gain of function will cause cancer. The next one is the tumor suppressor gene. We start with a 5, cell cycle control protein. So think about that, and you will see a lot of these. So here, a cell cycle control protein, which is a negative regulator. At one point, this cell cycle control protein is telling the cell that, okay, all right, there is some problem with the cell. You do not divide this cell. If it is a loss of function of that control protein, now the cell, without listening, going into the cell division, and a damaged cell is just accumulating, that can cause a cancer. That's your class 5, cell cycle control protein. Then we are going into the class 6. Now, this is a different group of protein, class 6, which are DNA repair protein. These are not essentially, they're grouped under the tumor suppressor genes, but they are really not directly under this class. And why it is so? So the DNA repair proteins directly are not involved into cell division or cell growth. But think about that while your DNA polymerase is replicating your DNA in the synthesis phase in the cell, it may make mistakes or mutations. So these DNA repair proteins will repair those mutations. These are exonuclease activity. They will repair those so that there is no damage into the DNA. So now these repair proteins, if you have a mutation, if they cannot, loss of function, remember that, loss of function of the DNA repair proteins will increase the mutations. And if these mutations are somewhere here in any of these proteins, that can turn on an oncogene, or that can loss of a function of a tumor suppressor gene. So that's basically what it is, that loss of function of a DNA repair proteins can lead to cancer as well. And that's the reason that as a loss of function, they are under this tumor suppressor gene category. And the last one is the apoptotic proteins that we did not talk about. So the cell do have a very specific, precise control that if you have unnatural growth or a cell is dividing where there is DNA damage, the cell do have a mechanism to program the cell to kill itself. That is what is called as an apoptosis or programmed cell death. So loss of function of these class 7 apoptotic proteins will also cause the cancer. Means that when the cell needs to be killed by the apoptotic protein, the mutation is not allowing the cell to be killed, and that can become a cancerous cell. So that's your loss of function. So this is the same questions. Don't worry. I just had only one question for today. That's it. So any questions so far? Any guys? Just wondering.
No questions.
No questions.
No questions. Everybody good?
Think so.
All good.
Okay. All right. I will, what's the time? 1:25. I will open up my office hours from 2 o'clock to 3 o'clock. So I will be there 2 o'clock to 3 o'clock. If you need to stop by, you can just stop by. You will see that my office room through the Zoom will be open. All right. So
Thank you so much, Professor.
Yeah, sure. One thing I'm just wondering that what I will do that next week will be the last week. I will open up your exercise or the for these two sections by this weekend. So you will be, even if we just do not finish the part 2 and 3, you will be able to see all the practice questions beforehand by this weekend. So you will have a whole 1 week of buffering time. So I will just upload all these so that you can just, again, start working on them. Good? All right. You folks have a good weekend. Just stay safe.